Jacob and Monod discovered the function of the repressor through a series of lacI mutants. And recall that the wild type has inducible expression, and what that means is that there is no beta gal or permease expressed when you don't have IPTG or you don't have lactose. When you have lactose, then both beta gal and permease are expressed, and that's referred to as being inducible, where the addition of IPTG or lactose can induce the expression of beta gal or permease. The first mutant Jacob and Monod looked at was I minus, and what they discovered was that it was a constitutive mutant where beta gal and permease were expressed irrespective of whether lactose is present or absent. And let's see how this might work mechanistically. So if you have a mutant lacI, that means it does not make a functional repressor protein, and that implies that the operator is not bound by repressor, and therefore the polymerase can bind the promoter and elongate, and we will get expression in when you don't have any lactose. You will still get expression of Z and Y, leading to constitutive expression of beta gal and permease. Next, Jacob and Monod investigated the dominance relationship between I plus and I minus by constructing a heterozygous partial diploid. And when they did so, they found that the I plus allele was dominant because the expression was inducible rather than constitutive in this I plus over I minus heterozygote, and that leads to the conclusion that I plus is dominant over I minus. Let's see how this might work mechanistically. So we have I plus on the chromosome and I minus on F plasmid, O plus on both of them, and Z plus and Y plus on both of them. Now we have two different alleles of the lacI gene. I minus does not make any functional repressor protein. However, the I plus allele on the bacterial chromosome will make a functional repressor protein, which is then going to go and bind to the operator sites on both the DNA molecules, so both on the bacterial chromosome as well as on the F prime plasmid. And in the absence of lactose, this will prevent the repressor bound to the operator sequence, will prevent polymerase from binding, and we will have no transcription, and that's why we get no beta gal and no permease expression when you do not have any lactose or IPTG in the cell. Now, when you do have lactose, this repressor is going to undergo an allosteric transition due to binding by IPTG or lactose, and then lose the ability to bind to either operator site, allowing polymerase to bind the promoter and do elongation, resulting in the expression of both beta-gal and permease, and overall this will result in an inducible pattern of expression as observed by Jacob and Monod. Next, Jacob and Monod wanted to investigate whether, similar to OC, the mutant for the operator, I minus acts in cis or not. And they were able to achieve this by pairing the I minus allele with Y plus and the I plus allele with Y minus. And the prediction, if I minus acted in cis, was that the is that the permease should show the I minus phenotype, and that means the permease should be constitutive, whereas beta-gal should show the phenotype of I plus, and so beta-gal should be inducible. But instead, what they observed was that both beta-gal and permease were inducible, and permease was not constitutive as would be expected if I acted in cis in a manner similar to the operator. And now let's see how this might work mechanistically. And so my I minus allele does not give any functional repressor protein, whereas the I plus allele gives a functional repressor protein. Now, when we don't have any lactose in the cell, then this repressor protein can diffuse and bind both the operator sequences. Now, recall that this is a partial diploid cell, and so these two pieces of DNA, one which is the bacterial chromosome and the other which is the F prime plasmid, are part of the same cell. And even if the repressor protein molecule is made by one of the DNA molecules, in this case the F prime plasmid, these repressor protein molecules can diffuse, can move around in the cell and go and bind to the operator sequence of the other allele or the bacterial chromosome. And as a result, you are able to block the polymerase from binding the promoter, and there is no transcription, resulting in no beta-gal or permease expression in cells where there is no lactose, even though the I minus allele is linked to Y plus allele. And now if we have lactose in the cell, then the repressor molecule will undergo the allosteric transition and not lose the ability to bind to the operator sequence, and since it's the protein that has changed, it will bind neither the operator sequence on the bacterial chromosome nor the operator sequence on the F' plasmid, allowing polymerase to bind the promoter and carry out transcription on both the alleles on both the bacterial chromosome as well as the F' plasmid, resulting in expression of Y or permease and beta gal. And this the end result is that expression is inducible rather than constitutive. And the conclusion from this experiment is that I minus acts in trans. So in cis means on the same site, on the same DNA, and in trans means across or from different molecules of DNA. A final mutant that Jacob and Monod looked at was IS. And when they looked at the phenotypes in IS, they found that not only were beta gal and permease not expressed in the absence of lactose, but they were also not expressed in the presence of lactose. And this IS mutant provided evidence for allosteric or the allosteric transition that the repressor undergoes when lactose is present, because evidently the repressor is acting as if there were no lactose, even though lactose is present. And let's see how that might work mechanistically. So I have IS and O plus, Z plus, Y plus. Now the IS mutant makes a repressor, a mutant repressor whose shape is different, so that the allolactose or IPTG cannot bind to it. And so we will say that this allosteric site where allolactose or IPTG bind to it, it's not available. Now, when you don't have lactose, then the repressor goes and binds the operator site, prevents polymerase from binding the promoter, resulting in no transcription and therefore no beta gal or permease when you don't have any IPTG. The difference here arises that when you do have lactose, because of the mutation in the repressor that's made by the IS allele, lactose is not able to bind the repressor and change its shape, preventing it from binding the operator sequence. And so now this IS, the repressor that's made by IS, will go and bind the operator sequence even though lactose is present, preventing polymerase from binding to the promoter and elongating, and that is what results in no beta gal or permease expression even though lactose is present. So this is the opposite of inducible. It is constitutively repressed And this mutant is called the super repressor, since it is able to repress in the condition where lactose is present, where the wild type repressor is not able to repress transcription. Jacob and Monod also investigated the dominance relationship between the super repressor IS and the wild type allele I plus by making a heterozygote partial diploid. And what they found was that expression was repressed in all the conditions, and that implied that IS is dominant over I plus. And let's have a quick look at how that might work mechanistically. So IS will make a mutant repressor protein, and I plus will make a normal repressor protein, and either of these can bind the operator when no lactose is present. It doesn't matter which one binds, or both take turns binding. Either way, they will prevent the polymerase from binding to the promoter and transcribing, and that's what will result in no expression in the condition when you don't have lactose or IPTG. Now, when you do have lactose, you will have the two species of the repressors, and the wild type repressor will undergo the allosteric transition, and this molecule will not have the ability to bind the operator sequences. However, the super repressor, which does not go an allosteric transition because it has lost, the mutation has caused it to lose the ability to bind IPTG or allolactose. Now the super repressor will go and bind the operator on both the DNA molecules, the chromosome, as well as the F prime plasmid, and this will prevent polymerase from binding to the promoter and carrying out transcription, and that is what results in no expression of beta-gal and permease even in the presence of lactose, whereas normally it's inducible and you do get expression of beta-gal and permease. To summarize, the genetic analysis of these mutant E. coli strains in combination with genetic techniques like partial diploids and the use of IPTG allowed Jacob and Monod to discover how the lac operon is inducible, which is it is not expressed when there is no lactose present, but is induced and is expressed when lactose is present.